Hi guys, we're going to be learning about mutations today. So we're kind of learning how when anything is either added or taken away or substituted into the DNA, how that's going to change your protein and therefore your phenotypes. Okay, so the standard is describe gene chromosomal mutation, so describe the mutation in a DNA sequence. You need to be able to analyze the base pair in either a point or frameshift mutation. You'll learn those two terms today to determine if there's a phenotypic change. Phenotypic, just think physical traits. Phenotype, physical traits. So how your hair changes or your height or anything like that. Okay, so the definition of a mutation involves changes in the sequence of nucleotides. Remember, your nucleotides would be, think about like your bases. So like adenine, thymine, guanine, cytosine, uracil to the RNA. So because it will change then too, or changes in the number of the structure of chromosomes. So also the number of chromosomes can get wacky as well. Point mutation. So this is a change in a single base pair and occur at a single point in DNA sequence. It involves a substitution in which one base is changed. So this is a great example. 1 to the 5th base, which is originally, the original sequence had thymine. During DNA replication, it changed to cytosine. Now remember, every 3 bases will eventually code for an RNA, and then it will then code for an amino acid. So silent mutation, it doesn't change anything. So even though you change a base, it codes for the same amino acid. As you guys remember in that chart, the same base could code for the same amino acid. So there's nothing, so you don't even know about it, right? Like there's no effect on that person. Here's another example. So even if you go, this is the DNA sequence, not RNA, but if it was CAA and it changed to CAT, which by the way it is, so it would still have the same amino acid, so there's no change. Silent, you don't know about it. A nonsense mutation is the mutation that it causes a change. So an example is if you had AAA, if that coded for UAA, it would tell it to stop instead of having lysine as the amino acid. And translation would end, so no other tRNA would bring in any other amino acids, and now your protein is defective because it's too short. Okay, so the other term is frameshift mutations. So your point mutation happens at one point. Frameshift can be one or more bases. It's either inserted or removed from a DNA sequence. The first one of these is called substitution. You're substituting. So it's, if you think about the terms, it makes sense. One base will replace a different base. So this is like your base, right? These are your bases, C, T, G, G, A, G. And it gets changed to C, T, G, 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 G. And this happens during DNA replication. An error occurs with the DNA polymerase. It put in the wrong base and it didn't check itself. The next one's called insertion. It's when an extra base is added. So if you take a look at this, this is like your starting sequence. An extra adenine and thymine, 2 of them. That's why we're in this one. Is added in, right? So now what happens? Like now at your every 3, you've got extra bases in there, and now when it gets transcribed and translated, you end up with a different amino acid. So insertion. Deletion, a base is taken out of a sequence. So you can see here it was AAT, GGTG. If you look here, they took the T out, and now you're missing one here, and now you have a completely different sequence. So it's going to code for a completely different amino acid. So causes, what causes these things? It's either natural. So if it's natural, that means it happened either during DNA replication or during transcription. So either when it's going from, it's taking one DNA and replicating it, or like during mitosis, or during transcription, so when you're making from DNA to RNA. That's a natural process, and there's nothing that we do. Mutagens. Mutagens are environmental factors. These are things like pesticides, tobacco smoke, environmental pollutants, nitrates from hot dogs, really bad. There's things like in our makeup for girls. There's infectious agents too. I didn't put that on here, but like HPV, any bacterial. So anytime you get like a bacterial infection, it can also change your DNA. So you have physical mutagens. Physical mutagens would be like an electromagnetic waves, such as X-ray and ultraviolet rays. That's why they put something on your organs, like your soft organs, when you get a X-ray to protect those in ultraviolet rays. So the harmful effects. The harmful effects is, so how is it bad for you? Like, how is a mutation bad? It's going to change the protein structure, and it causes you to disrupt normal biological activities. Some cancers are a result of a mutation. An example is xeroderma pigmentosum. It's when there's a protein that usually repairs DNA damage by the UV light, and you don't have that, so these people can't be out in the sun because any damage is not going to be repaired, and that can cause skin cancer. There are also some beneficial effects to produce proteins with new or altered functions that can be useful to organisms in different and changing environment. For instance, this is not good for us, but it's good for them. African mosquitoes have created resistance to chemical pesticides. Plant and animal breeders make good mutations. So that's one example. So like this species has created a mutation so that they don't die from pesticides that they're immune to. Again, not good for us, but good for them. Plant and animal breeders make good mutations, giving an organism extra sets of chromosomes. It's called polyploidy. And basically what that does is it makes them bigger and stronger. So like when we import plants like bananas, limes, and strawberries, they produce them this way so they can last longer when they go overseas or anything like that. And yeah, so like your flowers will be bigger or your fruits are bigger. Okay, so that's it for the Ed Puzzle today. Good job, guys.